prayer for frontliners from a grateful nation. Lord Jesus, our Savior, you praised the good centurion for caring for his servant. Wrap our frontliners with your love as they risk their lives for people they do not even know. You heal the hemorrhaging woman by allowing her to touch you. Give our frontliners your peace as they touch so many frightened lives. You brought back to life the daughter of Jairus and returned her to her parents. Assure our frontliners the joy of returning to their loved ones as they serve you and save our nation. Already our transformation is happening as we witness their selfless dedication. Divine Healer, may their courage to face danger be not in vain. Please bless the work of their hands, that together with them, we may all know that you are God. Blessed Mother, our frontliners are precious to you as they are to us. Let them rest on your lap when they are tired and find comfort in your embrace when they are weary. We ask all these and trust in Jesus' mighty name. Amen.
Mga kababayan, ang pambansang awit ng Pilipinas. Kayang magiliw, kaya sa silanganan, alaw ng puso, sa nitip mo'y buhay. Lupang binirang, kuya ka ng magiting, sa mandulupin, di ka pasisigil, sa nagagang tulong sa simoy at sa langit kong pagaw, ay hiragang tulakaw, sa pagkayang minamahal, ang isang ng patawad mo'y tagumpay na naghihingi, makituwid at karoon na kailan, pangahirap tinitindig, luman ang araw ng warhabis magsikra, buhay langit sa mo. Ang hindi kaya na pagkay pangahit, ang mamatay ng dahil sa'yo.
Before we start our webinar, here are some reminders and guidelines for our virtual participants. First, please observe proper webinar and video conferencing etiquette. Also, please change your settings to speaker view. We are also kindly requesting all participants to turn off their video. You may post your questions regarding the lectures through the link flashed in your screen. Alternatively, you may also post your questions through the chat box in your Zoom console. Lastly, details on how to get your e-certificates will be flashed at the end of the webinar. We expect your adherence to these guidelines. Thank you very much. The Biorisk Association of the Philippines, in collaboration with the Philippine Association of Medical Technologists, warmly welcomes all our virtual participants to our 4th joint webinar. This webinar will focus on biosecurity, the awareness of laboratorians in biosafety and biosecurity measures, particularly during this time of pandemic, shall be highlighted for them to be alert in recognizing biosecurity threat. The new technologies and the dual-use research of concern will be discussed, and participants will learn why it is important to know how to identify it, what is the role of risk-benefit assessment, and how to develop a risk mitigation and communication plan. Let us all welcome our session moderator for this webinar. He is an active BRAP member, a practicing veterinarian, and associate professor from the College of Veterinary Medicine of Central Mindanao University. Ladies and gentlemen, Dr. Jose Obedencio Jr.
Good evening, everyone. Once again, welcome to the 4th webinar of the Biorisk Association of the Philippines in collaboration with the Philippine Association of Medical Technologists on Laboratory Biosecurity 2020, a focus on emerging biotechnology and dual-use research of concern. Tonight's session is viewed by our official participants via Zoom, and we also have our viewers through Facebook that is currently streamed live via the Facebook group of the BRAP. To formally open the event and welcome you all, I give you the National President of the PAMET and Vice President of BRAP, Mr. Ronaldo Puno.
A very good day and a biosafe and biosecure greetings to all our participants joining around the world. It is my distinct honor and privilege to represent both the BioRis Association of the Philippines and the Philippine Association of Medical Technologies in welcoming you to the fourth joint webinar entitled Laboratory Biosecurity 2020, a focus on emerging biotechnology and dual-use research of concern. Laboratory biosafety is more than just safeguarding of dangerous pathogens and toxins from individuals or organizations who would use them for harm. While protection of dangerous pathogens and toxins is obviously appropriate, the scientific, medical, and pharmaceutical communities should also consider protecting materials with historical, medical, epidemiological, commercial, or scientific value. This decision should be taken with due consideration to the fact that scientists serve only as temporary custodians of valuable scientific assets whose past and current value to science may be understood, but whose utility for the future can only be estimated. Dual-use research of concern is life sciences research that is intended for benefit, but which might easily be misapplied to do harm, and SARS-CoV-2 is not an exemption. The advances of science open doors to infinite possibilities to make use of acquired knowledge and techniques. Systems and controls should be in place to avoid illegitimate or unethical research. Researchers, laboratory workers, and biosafety and laboratory biosecurity managers should communicate and collaborate and strive to find the correct ethical balance for the activities performed. The possibility that dual-use research might result in misuse, either intentionally or accidentally, is a long-standing concern of science. The issue are broad and encompass not only research and public health, but also security, scientific publishing, and public communications, biotechnology, and ethics, and wider societal issues. So, let's take this opportunity to learn more and be more responsible in the use of research as we aim towards improving life through life sciences. Have an enjoyable webinar and good day to everyone. Thank you very much.
Thank you so much, Sir Ronnie. And now, tonight's event is quite a long session because we will have three speakers. So we will proceed to our first talk on a decade on biosecurity in the Philippines. Our speaker is a physician pathologist by profession who, after finishing anatomic and clinical pathology at Makati Medical Center in 1998, chose to shift career to medical research at the Research and Biotechnology of St. Luke's Medical Center in Quezon City in 2001. He co-founded the Philippine Biosafety and Biosecurity Association Incorporated in 2008. Meanwhile, taking the U.S. Department of State-funded Advanced Biosafety Officer Training course, finishing as Batch 1 in 2011. With his training and background in biosafety, he was appointed as Biosafety Officer of St. Luke's Medical Center in 2013 in Quezon City and in Global City in 2015. Seeing the tremendous risk laboratorians were facing with the gamut of pathogens in the laboratory, he called PAMET to join with him in embracing biosafety and biosecurity. which PAMET unconditionally accepted. It was then that he founded the Biorisk Association of the Philippines, Incorporated, also known as BRAP, in 2015, with majority of his founding officers were medical technologists. His efforts to advocate biosafety in the country, despite the hurdles and walls he needed to climb, he successfully overcame all this and caught the eye of the International Federation and Biosafety Associations in Ottawa, Canada, and awarding him the title IFBA Biosafety Hero in 2016. He sought professional certification with IFBA in biorisk management and biosecurity and obtained this in 2017. Without further ado, help me welcome Dr. Miguel Martin In Moreno II, the BRAP founding and incumbent president.
Good evening, everyone. I will present here tonight my experience on how BRAP and I, together with PAMET, advocated laboratory biosecurity and biosafety in our country. Let me share my slides. It's okay?
Yeah, it's okay, Doc.
Oh, thank you. It doesn't like to move. I will shut off my video so that I think it okay, can you hear me?
Yes, Doc.
Okay, this presentation will focus on my involvement in its advocacy on how laboratories in the Philippines came to know about biosafety and biosecurity in the last 10 to 11 years. This presentation is my own creation and does not mirror the goals and objectives of PAMET and or BRAP. I personally learned all about biosafety, biosecurity, and then biorisk management way back in 2008 when the laboratory I was working with recommended me to join a training funded by the U.S. Department of State Biosecurity Engagement Program. It doesn't like to move. Okay, so these next two slides, this is the first one, are my first original slides way back in 2008 that I used to present to participants to show definitions and key terms I use in the course. To review, biosafety is about prevention of unintentional risks or protecting people from pathogens, while biosecurity is focused on preventing intentional risk or protecting pathogens from the people. In addition to protecting occupational workers, laboratory biosafety safeguards the public and the environment from accidents involving pathogens and toxins. Bioscience, laboratory personnel, and management have a similar responsibility to implement biosecurity systems, but the concepts of biosecurity are much less understood than biosafety. The word laboratory is used in front of biosafety and biosecurity to help guide the definition and avoid confusion with other meanings of the words biosafety and biosecurity. However, biosafety and biosecurity principles apply to situations outside the laboratory as well, such as field work or in the transport of biological materials. The term biosecurity has multiple meanings or definitions, and this can lead to confusion among participants. To make sure everyone is on the same page, it is important for us to clear about which meaning of biosecurity we are referring to in this webinar. This definition was lifted from the 2006 edition of the WHO Biosecurity Guidance, and it states protection, control, and accountability for valuable biological materials within laboratories in order to prevent their unauthorized access, loss, theft, misuse, diversion, or intentional release. Here's the official WHO publications showing the evolution of the first edition in 1983. I have not seen the hard copies of the 1983 and 1993 editions. The very first edition I got my hands on was the 2003 edition. I have witnessed its evolution and improvement since then. The 2004 edition was used for a very long time until the fourth edition came out in 2009. And laboratory biosecurity, as defined in the fourth edition, refers to institutional and personnel security measures designed to prevent the loss, theft, misuse, diversion, or intentional release of biological agents being handled in the laboratory. A little thoughts about laboratory biosecurity as being emphasized in the fourth edition. Addressing laboratory biosecurity risk in many ways parallels and complements that of biosafety risk management. Effective biosafety practices are the foundation of laboratory biosecurity, and biosecurity risk mitigation must be performed as an integral part of an institution's biosafety program management. There are many challenges and caveats associated with implementing biosecurity policies and procedures. For example, many biological agents against which protection is needed can be found in their natural environment. And in addition, biological agents of interest may only be used in very small quantities or may be capable of self-replicating, making them difficult to detect or quantify. In some cases, the process of quantification may itself pose biosafety and biosecurity risks. Furthermore, while there is potential for malicious use of these biological agents, their use is valuable for many legitimate and benign diagnostic, commercial, medical, and research applications. For this reason, it is essential to properly assess potential biosecurity risk and establish appropriate mitigation measures that can reduce risk without hindering scientific processes and progress. These measures should comply with national standards and regulatory procedures and be proportionate to the assessed risk. To achieve this, an approach similar to the biosafety risk assessment framework should be undertaken with specific focus on biosecurity to identify whether an institution possesses biological agents that may be attractive to those who may wish to use them maliciously. The depth of this biosecurity risk assessment should be proportionate to the identified risk. For most laboratories, biosecurity risk assessment can often be combined with a biosafety risk management rather than being a standalone activity. As with biosafety, the biosecurity risk process also includes the development of strategy to manage the biosecurity risk by selecting and implementing biosecurity mitigation measures. A laboratory biosecurity program is required to prepare, implement, oversee, and review these processes according to the requirements of the facility. In many cases, this can be combined with biosafety program management, although it may need to be a standalone program where the biosecurity risks identified are severe and or numerous. I know you will recognize this figure. It was the one we started using way back in 2006. So for a short review, physical security, attending to access control, intrusion detection, alarm assessment, response, and then you have personnel security, background checks, periodic investigations, which has already evolved into something higher now, pathogen security, or otherwise known as material control and accountability, focusing on detailed inventory, internal, external transfer, and inactivation and disposal records of pathogens, And of course, transport security and the forever three-way packaging system, knowing from where, how, and where will it be transported. Having a knowledgeable IATA certified personnel on board always, and information security, which deals with confidentiality, integrity, and availability of information, and also passwords and backups. So these were the elements of biosecurity way back in 2006 when I started lecturing with PAMET and other associations. In 2014, after attending a conference in Europe, I came home with this information of having 8 pillars of biosecurity already, which I cascaded right away a day after I arrived. I think that was in PAMET Bulacan chapter. So we see the 5 pillars as described earlier, and the additional 3 pillars are organization management, awareness, and biopreparedness. Organization management is simply ensuring management and administration that they fully support the biosafety and biosecurity program of the institution by institutionalizing the program and finding resources for its program implementation and activities. Awareness is simply making sure that the biosafety and biosecurity program policies were cascaded to the entire institution from top to bottom and from left to right. Biopreparedness and incident management is preparing the laboratory against any form of lab activity disruption and how to respond to the disruption up to recovering back after the disruption is resolved, which was termed resiliency. In the new WHO Laboratory Biosafety Manual of 2019, they have 8 new pillars. In addition to the WHO has made big changes in this edition, where from the original 5 elements, inventory control, let me get that out. From the initial 5 elements, inventory control, information control, personnel control, physical security control, transport control, and the last is emergency incident response plan. As I said, WHO made changes where from the original 5 elements, the one element from the 2014 list, which was biopreparedness, they have added 2 new pillars, which are dual-use research of concern and emerging biotechnology. These 2 additions will be the topic of our next 2 speakers. Thank you very much for your attention. These are my references in case you would like to read on.
Okay, thank you so much, Dr. Martin. Thank you. For those who have questions, you can place it in the chat box. We will have the open forum after all the speakers have delivered their talks. So we'll proceed with our next talk on dual-use research and dual-use research of concern. Our next speaker is working as technical advisor at Health Security Partners, where she provides coordination and technical support to HSP's health security programs in Pakistan. She has 5 IFBA certifications and is serving as an IFBA Global Mentor. She has also worked as ASM Young Ambassador to Pakistan in 2013 to 2016 and HSP's Health Security Future Fellow in 2015. She has been providing biorisk management training to international organizations. Please help me welcome Dr. Samreen Sarwar.
Okay. Hello, everyone. Good evening. And I'm very thankful to BRAP and PAMET for the invitation to discuss DURC today. And in 2015, I assessed the level of awareness and attitudes of researchers in Pakistan towards dual-use research of concern through a survey. and published my findings. Since then, I have been doing training on dual use research of concern and updating my knowledge on it. So I know that this topic might be new to majority of you, so I'll welcome your questions and I'll try to answer your questions, as many questions as possible in this lecture, or maybe you can, I will be able to answer your questions through email after this lecture as well. So let's discuss why you need to know about DURC and what actually means by the term DURC. So I'm sharing my screen. I'm turning off my video so that you can, there is no internet disruption. Okay. So what is dual use research of concern? This is very important for all of us to know. What does this mean? Because this is a new concept and it came in early 20,000, but this concept is still new among many of us. And we need to understand this, why it is important and why it is relevant to all of us. In 2001, there was a group of researchers who constructed, like they made a polio virus using the nucleotide sequences which are present in databases on the internet. And what they did, they took the sequence from the database and they synthesized a polio virus from scratch in their lab. They synthesized that polio virus from chemically synthesized oligonucleotides. And when they published these findings in 2002, this sparked a huge debate in the scientific community that why this experiment was needed at its first place. What was the need to manufacture a polio virus in the lab? Because possibly this could be a recipe, or you can say this could be an idea for people with bad intentions, that similarly like polio virus, many other viruses can also be synthesized in the lab. And these viruses could be very good candidates for biological weapons. So this was very important to understand that what are the dual use consequences of this research. The researchers who did this research, they defended themselves by this saying that we want to test the possibility that is it possible to construct a virus in lab. And they also defended themselves by saying that if this virus, polio virus, is present all around the world, then why they will feel the need to synthesize it in lab. So this doesn't make sense. So but this sparked a huge debate in the scientific community. Later on in 2005, there was a research in which they constructed the influenza virus, Spanish flu. Many of you might know that Spanish flu came in 1918, and this pandemic stayed in the world from 1917 to 1920, and 50 million people died in this pandemic, which is 3% of the world's population at that time. So they reconstructed that virus in their lab. And this reconstruction again got many questions on it, that why it was important to construct this deadly virus in lab. So the researchers had said that if we will not prepare, if we will not study this virus, we will not be able to make countermeasures, we will not be able to make vaccines, we will not prepare ourselves for future similar pandemics. That's why it is important to construct this virus in lab and to study it, to explore the pandemic potential of this virus. So you see that biological research, there are both peaceful, can also be used for peaceful purposes, and there are some harmful, this can also be used for harmful uses as well. So what comes under harmful use? It can be used by bioterrorists or other people who have bad intentions. They can be used as bioweapons. And what are the peaceful purposes? They can be used in pharmaceuticals, vaccines, and diagnostics. So this is the dual use component of biological research. Early on, it was there was a concept that dual use is only dual use research is a limited group of research projects. Now, with the passage of time, people have, and the scientific community has started thinking that dual use research is not only a limited number of research projects are not only dual use research. Possibly all biological research can at least theoretically be dual use. Because, for example, if you are doing a research, you are studying just the pathogenicity of the virus, or you are just studying whether it is resistant to certain antibiotics or not. These simple pieces of information, when they combine, that could generate an information which can be used by people with bad intentions. So now we can say that every research is possibly dual use. And this is why it is important for all of us to know about how we should be looking at our research projects, how should we have a security mindset to look at our research projects and ourselves decide what are the benefits of this research and how my research can be misused by someone else. Okay. So in this lecture, we'll see how to identify which research is dual use research of concern. And once we identify that this particular research project is DURC, which is dual use research of concern, what will we do about it? Well, how will we assess and how we will manage the risks which are associated with it? So let's start with the controversy, which again came up in 2011, that 2 manuscripts were submitted to Nature and Science. And these 2 manuscripts, in these 2 manuscripts, there was successful synthesis of a more deadly variant of highly pathogenic influenza. And what was the most worrying thing about it was that this influenza had 60% mortality rate. So you can see that Spanish flu has only 2.5% mortality rate, but this avian influenza had 60% mortality rate. And this was also able to transmit between mammals through airborne route. And this sparked a huge debate, and Nature and Science recognized the dual use potential of this research. And they said why it is important to do this research, why you feel the need to make a more deadly variant of highly pathogenic avian influenza. So the scientists said that we want to see what is possible, rather than just sit and wait how natural mutations which are occurring, they can possibly generate a more avian influenza strain which can spark a global catastrophe. So this is how the scientists justified this. So you can see that since early 2001, there have been a lot of research projects which came up, which has dual use potential. But how we will distinguish between dual use research and dual use research of concern? So dual use research is simply a research project, any research project which is done by a researcher for good purposes, for beneficial purposes. But there can be some risks associated with it, not biosafety risk. There are risks that it can be used by someone else, not by the researcher who is doing this research, by someone else using the information, using the product of that research, using the technology which is being generated as a result of that research. He can misuse this information, this product and this technology. So there are both uses. There are peaceful uses and there are harmful uses of one research. But when we will call this research as dual use research of concern, there are 2 main points on it. So how WHO defines it. So it's important that you understand the basic concept that what is dual use research. Biological research which can have both peaceful purposes and which can be used for bad purposes as well. But how we will define dual use research of concern? This is research, WHO defines this, that is intended for benefit. Like the researcher is doing it for benefit, but it can be easily misapplied to do harm. Okay, so let's see how NSABB defines this research. There are 2 important points. One is the scope, and the second one is how immediately it can be misapplied. So whenever we designate a research as dual use research of concern, so the word concern is important because it means that it has a huge scope of broad potential consequences or threat to public health and safety, to crops, to plants, to animals, to material, to environment. So it means what is the scope? It should be has significant threat to public health and safety, and it should be immediately misapplied. It should be able to be immediately misapplied. Okay, so when we see at the definition of DURC, any research project which can be easily misapplied, immediately misapplied, and has a broad significant threat to public health, means its scope of destruction is very high, then we will call it as DURC. Okay, so this is the difference between DUR and DURC. When the word concerns attached with it, it means it has a huge scope of catastrophic destruction, and it can be immediately misapplied. You do not need a lot of information, a lot of support in terms of like all the information is readily available in this research project that it can be misapplied to do harm. For example, if we take the first example, in our first example, we saw that they make a highly pathogenic avian influenza virus strain. If that strain is released into the environment, it can immediately cause major public health. It can threaten public health and safety. It can cause a lot of deaths immediately. Okay, and its scope to cause deaths is very high. That's why we can designate that research as dual use research of concern. So when the experiments, there were controversial experiments when they start coming up in 2001 and then 2 and then 3, National Research Council in US, they made a committee which was chaired by Fink. Okay, so they made a committee in 2004, and all the scientists came together, they sit together, and they saw that we should do something about it before these research projects are being published openly, and anyone can access this information and can use it for bad purposes. So these were a few they made a Fink report, and the name of this Fink report is Biotechnology in an Age of Terrorism, Confronting the Dual Use Dilemma. So they made this report, which is called as Fink report. And what were the recommendations of this Fink report? They said that we should educate the scientific community, national and international organizations. They should educate researchers about dual use research of concern, because ultimately the researcher is the one who knows his research the best. The researcher is the one who decides to do this particular project. And it is very important to educate that scientist. If that scientist is able to identify DURC, he will be able to report, he will be able to take proper mitigation measures from the very start. It's very important to educate scientific community. It's very important to educate other stakeholders. Okay. Then the second point is review of plans for experiments. So what does this mean? That, for example, IRE, which is institutional review entity, or it can be make part of institutional biosafety committee. Whenever the research project comes to IBC for approval, they should have DURC experts on board who can evaluate that research project, whether it has any dual use potential or not. And if there any DURC potential comes up, they should address it. They should ask researchers to take proper mitigation measures or modify their research. Then publication stage review tells us that the journals which review the articles and which publish the articles, they should have a DURC, they should review it from the DURC perspective. Okay, so if, for example, in our earlier example, we saw that in Nature and Science, they themselves reported, they came up that this research has dual use potential and it should be taken care of. Okay, so this is very important that the journals should have proper people who have the expertise in DURC, and they should evaluate research projects on DURC grounds. Then they feel the need of creation of a National Science Advisory Board for Biosecurity who can provide advice or dual use research to both government and to the researchers. In addition to this, There should be improved communication between law enforcement and life science organizations, because ultimately if you are going to report something, you'll have to both of these stakeholders should understand each other. So otherwise it will not be able to properly highlight these problems and report these problems. Sixth point was that, so for example, a researcher made a very highly pathogenic avian influenza virus in their lab, but not intentionally, but unintentionally, this virus was, if it is released into the environment, it can cause similar damage of broad potential consequences. That is why it is also important to have proper physical containment, and they should sort out the biosecurity and personal issues. And then there should be international oversight, because because the publication goes everywhere, anyone from around the world can access this publication. It means they can use this information. If there is no coordination among internationally, then it will not work. There should be consensus. There should be internationally there should be consensus between countries on how to regulate and how to do proper oversight of this kind of research. Okay, so now we will discuss what are the 7 categories of experiments. So NSABB, which I told you that they created a National Science Advisory Board for Biosecurity. They said that what we can do, we can identify some categories of experiments which has high potential that they fall under DURC. They have high potential that they can fall under DURC. So they identified 7 categories. These 7 categories, for example, if you are enhancing the harmful consequences of a biological agent, for example, you are making it more virulent, or you are making it more like able to infect people with more virulent, and it has more consequences after infection, or disrupting immunity. For example, you can make a virus which is against which the vaccination which is already in place will not work against this virus. Okay, confer resistant. For example, if you have, for example, doxycycline for Vibrio, making strains which are resistant to antibiotics or therapeutic interventions which are already in place, or you are increasing their stability in the environment, you are increasing their transmissibility for any reason. For example, there was an experiment on Clostridium botulinum, and the purpose of that experiment was to make more resistant spores of Clostridium botulinum. And the reason behind that was that these spores will not be able to study these spores in detail if you will not make it more stable. So that's why they made their spores more stable. But that was not for any bad intentions. That experiment was for good. But ultimately, you are giving a recipe on how to make Clostridium botulinum spores more stable. So that is why it comes under the DURC, because you are increasing the stability, increasing the transmissibility. Or you want to alter the host range, like for earlier on it was not able to infect birds, now it is able to infect birds. For earlier on it was not able to infect mammals, now it is able to infect mammals. Or you increase the susceptibility of a host population, like for example, you enter a gene into a virus that whenever they enter into the body, the immune system will not work as a result of that gene. Okay, or you are generating a novel pathogen, or constituting an extinct biological agent. So these categories, all 7 categories of experiments, they can come under DURC. They don't come under DURC, they can come under DURC. We'll have to do assessment and we'll see whether they can come in DURC or not. Okay, so these are experiments of concern. So whenever these your research falls under these categories, your research will need identification whether this is DURC or not, and then it will require more concern, more vigilance, and more oversight. And then there are select agents. These select agents are because they are very the US name of these agents is select agents. They are also called as security sensitive biological agents. They are also called tier 1, tier 2 agents, or specially dangerous pathogens. There are different names for these sort of agents in different countries. Why these agents are important? Because they have high potential to be misused in as a biological weapon, because they have like all the properties which a biological weapon should have. Okay, so now after knowing the 7 categories of experiments and after knowing the select agents, now we will see how will we identify that our research is DURC or not. It doesn't mean that your experiment falls under 7 categories that it is DURC. It doesn't mean that it is DURC if your experiment falls under 7 categories. If you are working on a select agent, it doesn't mean that your experiment is DURC. To be called as DURC or to be designated as DURC, that particular research project should fit into the definition of DURC. If it doesn't fit into the definition of DURC, whether it is one of the 7 categories of experiments, whether it is select agent, it will not be called a DURC. Okay. So to be called as DURC, it should fit into the definition of DURC. Okay. And there is another case as well. Sometimes you are doing research which doesn't fall under these 7 categories. You are not working on select agents, but still that research has the potential to be called as DURC. For example, in early 2001-2, there was experiments in which they were making a device which transmit aerosols to deep into the lungs. So they made it for asthma patients to deliver drug deep into the lungs. But they came up that the same device can be used for the spread of pathogen as well. So it doesn't come under 7 categories. It doesn't involve a select agent, but it fits into the definition of DURC. That's why it will be called as DURC. Okay. So once you designate that your research is DURC, and I earlier mentioned that to be called as DURC, you should have that particular research product should have the scope of broad potential consequences or significant threat to public health and safety. And the second thing was it should be immediately misapplicable. Okay, so it should be immediacy. So what are the two main things? It is the scope and immediacy. Scope and immediacy. So to be called as this, if it has the scope and it can be immediately misapplied, it will fit into the definition of DURC. Okay. Now, what you will do about DURC, if you have identified that your research project is DURC, it doesn't mean that research should be prohibited or should not be published. Okay. What you do is that if you, because we cannot hinder scientific progress, we cannot scientific progress just because of a small chance that terrorists can use this information to make something very dangerous for public health and safety. What we will do, we will apply proper mitigation measures. Why we want to identify DURC at the first place? That we will be able to apply mitigation measures. We will be able to see how it should be communicated. And if there is no benefits associated with this research, what we will do, we will discontinue the research, but this will be the last option. DURC designation doesn't mean that you stop research. You just, what you do risk and benefit assessment. So this is the question, that should scientists stop the research once it is designated as DURC? No, scientists should not stop the research. They should evaluate it. They should see what are the risks associated with this research. They should see what are the benefits which are associated with this research. For example, what are the risks? For example, biosecurity risks, there are biosafety risks, there are risks which are associated with communication. It can cause public threat, like it can cause a panic in the public. It can shatter public confidence. So these are all associated risks. It can be used for bioterrorism. These are the risks which are associated with DURC. But what are the benefits? The same research can be used to make, for example, as I earlier mentioned, that to make a spore more stable. So if you make a spore more stable, you will be able to study it. Okay, so what are the benefits? It will progress science. What are the benefits? It might be used, the information which will be generated as a result of this research, it will be used for therapeutic purposes, for vaccination, for other beneficial purposes. Okay, so then comes the question that after a careful risk benefit assessment, that once a research is designated as DURC, you assess the risks, you assess the benefits, then you weigh them. If the benefits are more than the risk, what you do? You will focus on the risk, apply proper mitigation measures, and you will try to bring these risks under acceptable range. Okay, and you will continue your research. You will do continuous monitoring of your research. You will plan out accordingly. You will apply proper mitigation measures, and you will see how will I communicate this research. What should be my communication plan? But sometimes it happens, for example, in certain research projects, the risks are more than the benefits. The benefits are very few, but the risks are very high. So what will you do? You will either modify it a bit. If you are not able to control the risks with proper mitigation measures, you will try to modify your research. And if it is not possible to modify your risk and still the risks are very high, you may consider decontinuing it. Okay, this is the last option, that you decontinue. But initially you think what mitigation measures can be applied, what control measures you can need to be in place, how you will continuously monitor it, and how will you plan communication. Okay, now you have risk and benefit. You have identified DURC, you have done risk and benefit assessment, and then you see that the benefits outweigh the risk. Benefits are more and the risks are less. Then you will see that how you can move forward. For example, the benefits are prediction of pandemics. Like we see that they made a more virulent strain of avian influenza. So this was important to have to prepare for the worst case scenarios, or to see what is the pandemic potential of this virus. Or you want to understand host pathogen interactions, or you can want to make effective countermeasures. Or this is very important, that there is one school of thought that thinks that when the research is funded with public money, public has the right to know about it. So when there was the research about the highly pathogenic avian influenza virus was under review in Nature and Science journals, they said that we will publish it after omitting critical information, like critical information which can be misused, like material and methods. But later on, there was a huge opposition against this. The scientific one group of scientists, they think that how you can like redact some information. Who gave you the right to hinder publishing full manuscript? So later on, they took their decision back and they allowed full publication, because scientists think that if you'll not publish complete results, the other scientific groups cannot challenge that research, they cannot do it, they cannot see whether it was actually the case or not. Okay, and the risks are, for example, there can be accidental exposure or release through rigorous biosafety and biosecurity procedures. It can provide a recipe for bioterrorists. It can provide unnecessary experiments that do not have a health or societal benefit. It can create a novel pathogen. So when we see it in the context of COVID-19 during this pandemic, we can foresee that this pathogen, which if it is stored without proper biosecurity measures, these pathogens can be acquired by research groups, and these research groups can do certain types of research on this pathogen. But if they don't know what can be the implications of their research, whether it is possible to use this information, some other groups can use your information for bad purposes. So in the context of COVID-19, you need to understand that whenever you are starting your research, you should analyze the risks and benefits associated with it. And if your benefits are more than the risks, then you should properly address these risks in your publication, and you should publish it accordingly. Okay, so it's very important that whenever you are going to do any research, analyze it from this perspective, that why it is important. So it is very important to have a security mindset. Okay, so how you will communicate it? So communication is very important. It is one of the mitigation measures. So are you going to publish it fully. For example, you are going to include material and methods, or you want to omit material and methods. What should be the timing? You want to publish it immediately, or you want to publish it later on? For example, you are working on a pathogen whose vaccine is not available until yet. Publishing it, like publishing this particular pathogen, a more virulent strain of this pathogen, can be more disastrous at this point. But later on, when its vaccine is already available, then the risk associated with research automatically goes down. So timing is also very important. And extent of distribution is that up to how like which people you want to access your research. You want to do it open access, completely any person around the world can see the content, or you want to access your research by certain groups who are doing the relevant research only, and who apply through proper channel to get access. So this is responsible communication. Okay, so this is basically DURC. In the end, I will like to mention a very key important points, that the 7 categories of experiments, if your research falls under 7 categories of experiments, okay, it cannot be DURC. There is a possibility that it is DURC or not, but if your research falls under these 7 categories of experiment, it requires more oversight, it requires more vigilance, it requires more concern. Okay, but there are research projects which doesn't fall under these 7 categories of experiment, but they still are DURC. Right now, why it is needed? Because now it is considered that almost all research projects should be analyzed from the very first stage when students are designing their projects. We should build a security mindset in these students. Okay, like researchers think that whatever they are doing it for good, but they don't think that what can be the misapplications of my research. This building a security mindset among these researchers is very important, so they are able to analyze their research from that point, and they continually revisit their research, because they are the most responsible in this term, because they are the one who knows their research the best. Okay, so this is very important, that we build a security mindset in our researchers from the very early career in their in research. Okay, so thank you very much, and we'll address questions in the end.
Okay. Thank you very much, Dr. Samrin. Okay, thank you very much, Dr. Samrin. That was a very comprehensive discussion on dual-use research of concern. So you can still post your questions for Dr. Samrin and Dr. Martin in the chat box or in the link that was provided earlier in the session. So we'll now go straight to our last talk, which will be on Biosecurity 2020, Ensuring Biosecurity. For speaker serves as the Director of Scientific Programs at Health Security Partners, or HSP, which is a nonprofit organization based in Washington, D.C., and dedicated to building local capacity for health security globally. His training is in molecular biology and biochemistry, and he holds a PhD from Johns Hopkins University. After conducting bench research for a few years, our speaker, Dr. Prasad, moved to the field of global health security, working through a number of nonprofit organizations to strengthen laboratory and health security capacity. He has been supporting partnership to prevent, detect, and respond to emerging biological threats in the Philippines and across the ASEAN region. Dr. Prasad and HSP has been a partner of BRAP since the very beginning, and working on multiple efforts and strengthened biosecurity systems in the Philippines. With that, we welcome Dr. Prasad Kuduvali.
Thank you for that wonderful introduction, and I would like to say thank you also to Dr. Martin and the BRAP committee for inviting me to share my thoughts on emerging technologies and biosecurity. Let me share my screen and then we can get started. Okay. I think both the talks given by Dr. Martin and by Samareen were perfect introductions to the topic. And this is just going to be a continuation. I think Dr. Martin covered the basic elements of biosecurity really well. And then Samareen focused on dual use research of concern. And this is going to continue on research, but research using new technologies that are coming out and how they affect our perception of biosecurity and what methodologies we have to reduce the risk from these technologies. So I will start by telling you what these emerging technologies are and how they work. And I should tell you that I am not an expert in these emerging technologies. My focus area is on the biosecurity threats from these emerging technologies. So I too am learning as I go. Every week, there are new publications, there are new discoveries in this field, and then people like me who read it often think, wow, we had not thought about that. That poses a significant risk. What do we do about it? But then these emerging technologies are superbly exciting because they have the potential to grow the bioeconomy. They can bring jobs. They can provide solutions to infectious disease problems, and as well as chronic disease problems that we face. So there's a lot of potential for growth, and I'll talk about that. And then I'll talk about the threats from these technologies, and then mechanisms for addressing the threats and ensuring biosecurity. So there are many emerging technologies. Today, I will focus on synthetic biology. And I think Samareen started us off on that subject. And then I will talk about gene editing systems. I think a lot of you must have heard about CRISPR-Cas, and then gene drives. And by the way, these are not the only emerging technologies. There's also artificial intelligence. There is UAVs, lots of other technologies that are coming up very rapidly that could pose biosecurity risk. So basically, synthetic biology brings engineering and biology together to make new things for new use and new applications. And people have often called this the design and construction of new parts, devices, and systems, or the redesign of existing biological systems. And this is a quick slide that I have to acknowledge Dr. Ji Lun from National University of Singapore, who introduced this topic to me, and this was a great slide from him, which compares what engineering does when they build a machine. They start with the raw materials, and then they design, resistors, capacitors, logic gates, circuits, and then you have the machine with an application. And similarly, people in synthetic biology are looking to do that with the components of a cell. We have enough information now about nucleotides. Basically, we have sequence information. We have genome sequences for a large number of genomes. And we know how genes produce proteins, and then proteins direct biochemical reactions, and then there are networks and pathways that ultimately lead to some kind of an output from the cell. So how do we take these various components and build something that will be useful to us? That's the basic goal of synthetic biology. And there are many applications to synthetic biology. Samarine, in her talk, told you about how they synthesized the poliovirus, starting from pieces of DNA in the lab. And then they also did that for the 1918 flu. Those are examples of synthetic genomics, where you start from scratch and then you build a DNA molecule, and then you do what is called genome engineering. And others have also worked on developing a basic cell, a bacterial cell, for example, that can be used for synthetic biology. There are, of course, there are many applications, including cell factories, where you use the cell to produce drugs, or you produce chemicals that are of useful use, commercial use. You can also develop them for genetic circuits, so that you can control what are the outputs of the cell. You can use it as biosensors for detection work, and I will talk to you a little bit about that soon. There are many interesting applications, including COVID-19 detection. You can use them as therapeutic cells, essentially, gene delivery, etc., where you are able to change the course of a disease through engineering. And of course, bioremediation, which is of value to in the environment. So these are some of the applications, and this is why this is such a huge, big deal. Many countries, especially in the ASEAN region, have invested very heavily in synthetic biology. There are a lot of startup companies that are working on these kinds of solutions. And there's, of course, academic research that is supporting these companies. And it's estimated that the market will be something like more than 38 billion this year. So we talked about reconstituting viruses. Let me give you one more example. This happened a few years ago. Canadian researchers developed, they basically built the horsepox virus from scratch. And they did this, they were at a university, but they did this in using private funding. So in many countries, if you use private funding, then you don't have to do the kind of oversight that Samarine was talking about under dual use research of concern. So really, I mean, the first time that people heard about this research was when it was ready for publication. It had already been done. And this was of great concern because, horsepox is closely related to smallpox. And by doing this research and wanting to publish it, the researchers were basically providing a blueprint for people, for labs around the world to be able to synthesize the smallpox virus. And I think all of us know why that is a high-risk proposition. So there were many concerns here, including what's happening with private funding that is not reviewed by government agencies. For example, if this was a NIH, National Institute of Health in the US, funded project, then there would have been oversight mechanisms, there would have been review, etc. But none of that was required because this was private funding. And then the researchers themselves, they said, well, we want to do this because we don't have access to smallpox, because as you know, smallpox is only stored at the CDC in the US and at the vector lab in Russia. Those are the only two labs that are allowed to have smallpox. And so they said, we don't have access to that. And therefore we thought, let's start here and come up with new vaccines for smallpox that would be helpful in case there is misuse of smallpox. Unfortunately, that was not a good enough justification. If you, again, Samarine talked about risk-benefit analysis. And really, if you go in and look at this paper and you read the commentary on it, it's very difficult to justify what was done. But currently there is nothing in place that prevents something like this from these kinds of experiments from being conducted. I get a lot of interesting, when I talk about this subject, and I've talked about it in many countries, and I get a lot of interesting reactions. And most of the reactions come from senior researchers and senior members in government who either say, oh, this kind of research is not happening in our country, or they say, we don't think this research is valuable for our country, and so we will not allow it. But what we see is that there is actually, this synthetic biology is very popular among the next generation of scientists. And there are many things that are happening, like the iGEM competition, which I have highlighted in this slide. The International Genetically Engineered Machine Competition, which happens once a year. And it is open to undergraduates and graduate students from around the world who can submit ideas and then carry out the research. And then they go to the iGEM and present their research, and they can win awards and recognition. And so a lot of groups, university groups, are very interested in this. And this year it's going to be at the end of October, and I assume, of course, it's going to be a virtual one, this one, but these competitions are happening. And if you go and look at the iGEM website, you will see that there is participation from all around the world. So even though you may not be involved in this research, in this kind of research, or you don't know anyone who is, your students might come up to you and say, we want to participate in iGEM and we have an idea for it. And then you have to deal with this. In addition to iGEM, there's also what is called DIY bio, or do-it-yourself biology. And you can go to these websites and check it out. These are groups of people, citizen scientists, what we call, and these are groups of people who get together and they, do experiments in their garage or in their, they have a small setup, and they do biotechnology related experiments. And there's a very popular one called the Open Insulin Project all over the US and abroad, which is focusing on how do we make insulin so that we don't have to pay the prices that are charged by the large biotech companies. I went and looked this morning, and there was a very interesting group called Manila Biopunk, and they are a DIY group in Manila. To their credit, they are very security conscious and safety conscious, and they are very open on their website, and they say that they do not work with pathogens. But it's interesting to see that, as I do talks in different parts of the world, and then I look at the website regularly to see what new groups there are groups all over the world. And this is actually happening. We may or may not know about it. And the problem with something like DIY biology is that it's very difficult to regulate this. And it's very difficult even to educate these groups, etc., because you have to provide, first you have to find them, and then you have to get to know them and get their confidence. Only then can you do training, awareness building, and some, exercise some amount of regulatory control. Gene editing. This is a very popular topic, and this everyone, I would assume, has heard about CRISPR-Cas. And this is basically a system that exists naturally in bacteria, and it helps the bacteria fight off infection by bacteriophages. And I have simplified it here, and basically it consists of a protein that is called Cas9, and then which you can see here in beige, and then there is a guide RNA, or gRNA. And using these two components, you can find any location in the genome. Basically, you can recognize, you can read DNA sequence, and you can find the sequence that you are looking for in the genome, inside the cell. Once you find it, you can cut at a particular location. And then because the cell will try to repair the cut, and we know how repair works, we can then add a desired sequence that we want in place of the cut. And that, you know, in a very simplified way, is how gene editing works. So you have the ability to find any sequence that you want in the DNA. You have the ability to cut that sequence at a particular location, and then you are able to join it and insert a sequence that you would like in that place. So you can imagine, if there are mutations that are causing disease, you can repair it. And so this is why CRISPR technology is so exciting, and I will talk about applications in a minute. I have over here a link to a TED Talk by the person who discovered CRISPR-Cas, Jennifer Doudna. And it's a great TED Talk. And I know that, you know, for a lot of people, reading publications is not always top of your list. But listening to a TED Talk is always interesting and engaging. So go check it out. It's a great way to learn how CRISPR works and what are some of the concerns with CRISPR. CRISPR kits are available for sale from various companies, so you can buy that. There are also online resources for designing your CRISPR. So basically, you have to design the guide RNA in such a way that it will recognize the sequence in the genome that you are looking for. And then there is also a DIY or do-it-yourself bacterial gene engineering CRISPR kit that is available for people who are enthusiastic about trying this out. And basically, it goes in and makes a mutation in the bacteria in E. coli, non-pathogenic, that allows the bacteria to grow on a different media. And so that way, you have basically changed the genome of the bacteria, and therefore you're getting growth on a new media. So here's one application. There are many diseases, and I think people have made an inventory of about 100 diseases that are the result of genetic mutations, and one is blindness. And so again, there is now it's been FDA approved to do a clinical trial for a gene delivery, a gene editing system that goes in and changes the mutation and corrects it so that people are able to see again. Now that's a great example of the kind of applications that gene editing provides, kind of solutions. There's also a COVID-19 testing a diagnostic that has got the approval from FDA. And basically, because you can read the sequence, you should be able to recognize any virus or any bacteria or other pathogens. And so there are a lot of diagnostic uses for CRISPR. This gene editing technology is, people in plant, in the field of agriculture are really, really excited about it, because you have all kinds of applications where you can make disease resistant, drought resistant, increased yield, etc. of basic foods, grains. And so from that, even commercial crops, and from that, that will ensure food security, and that will ensure that we have the best plants and seeds available to propagate. And this has always been done by the plant community. They have always been looking for better qualities of rice, a better quality of maize, etc. But now they have a tool that saves them time, effort, and money. Livestock sector, gene editing is very big. And it's easier to do experiments in livestock. So now there are pigs that are resistant to major diseases, PEDV and PRRS. And these are, again, in the experimental stages at this point. but these pigs have been created, and they are resistant to the disease because the receptor, which is used by the virus, has been modified in these pigs. In chicken, this is not at the animal stage yet. This is in the lab stage, where they have modified one of the proteins so that the cell can be resistant to influenza. So you can imagine in the livestock sector, this is huge. If these kinds of animals are allowed and they are found to be safe, this could change, the livestock sector forever. But there are concerns with these kinds of things. When you put pressure on the pathogen, remember the 7 things that Samreen pointed out, the 7 areas of concern for research. One of those was change of host range. What if as a result of this, there are mutations in the virus that alter the host range, or that alter transmissibility or pathogenicity? So those are the kinds of concerns and questions that will have to be asked before these kinds of things are actually allowed in mass production for livestock. And of course, we had the worst case scenario happen when a Chinese scientist edited human embryos and mutated the CCR5 gene, which helps to make people resistant to HIV infection. And since then, this of course is a huge moral dilemma, because you can imagine all kinds of designer babies. You can also imagine all kinds of designer applications for military purposes, etc., that can be done if we go down this path. And therefore, scientists all over the world have called for a moratorium on germline editing in humans. Then, based on gene editing, there is something called a gene drive. Now, if you look at the left side of this figure, you will see that if you use CRISPR and you change one copy of the gene, or one allele, as it's labeled here, then normally over generations, it takes the pattern of normal inheritance. And all of you have studied this at some point or the other in biology and genetics. This is normal inheritance, where there's a limited probability of being passed on to the next generation. But then if you do a trick using CRISPR-Cas, what is called a gene drive, where you are able to modify both copies, then what happens is that over time, all of your progeny will have the mutation, will have the change that you have created. So this gene drives are a way to make population-wide changes. And I could go into this and explain in detail, but we don't have the time here. So I leave you with a very interesting TED Talk from Jennifer Kahn that describes how gene drives work. And this is very exciting for those people who are working on vector-borne diseases, because in the lab, they have been able to create a gene drive that basically leads to a population collapse of mosquitoes, where the female offspring cannot bite, and therefore they cannot produce eggs. And therefore, over a few generations, the mosquito population dies out. So this, this is still in the experimental lab stage, but people are very excited about this. So a couple of years ago, when there was a call for a moratorium on gene drives, several countries voted against it, because they are very much interested in pursuing these technologies to control vector-borne diseases, which cause huge devastation, particularly in sub-Saharan Africa. So this is the most important and interesting piece from my perspective on how do we control these technologies, and how do we mitigate the risks so that we can continue to work on these technologies and all the amazing discovery that we are seeing. So we don't want to stop science, we don't want to stop the economy, but at the same time, we can be safe and secure. So at the national level, at all levels, you need policy. There is currently no policy on this that is satisfactory anywhere in the world, but it's an opportunity for people to come together and talk about this and come up with effective policy. At the national level, there should also be a body that will review, approve, and provide oversight of this kind of work. Now, in many countries, especially in ASEAN, in Singapore, and in Malaysia, they have taken, as you know, in every country, there is a body, the National Biosafety Committee, etc., that looks at GMOs and reviews all GMO products. So that committee has taken over the research oversight, review and oversight functions in some countries. The problem is that committee has no experience in biosafety or biosecurity. They have always worked on GMOs. And so now they're trying to bring in biosafety and biosecurity experts in order to be able to review this kind of work, because ultimately all gene editing falls under genetic modification of one kind or the other. And there needs to be awareness raising at the national level. Institutionally, policies are required. Samreen mentioned this, and I will reiterate, there needs to be robust institutional biorisk committee. Everyone is used to institutional biosafety committee. And people are quite good at doing biosafety risk assessments and taking biosafety measures in many countries now, because biosafety has been around for a long time. But biosecurity is a different thing. And there isn't a lot of expertise, and people are frankly not very comfortable with doing biosecurity risk assessments or doing biosecurity mitigation. All institutions, of course, need a robust biosecurity and BRM system, biorisk management system, training, education, and a culture of responsibility. All of these things we have previously talked about. At the individual level, some countries have a code of conduct that they have developed for scientists so that they will comply with the policy, they will comply with the best practices. And also individuals, scientists, need to be very comfortable with risk assessment and mitigation. This is a difficult thing to learn, and this is a difficult thing to do. And for those of you who have attended biosafety and biosecurity talks, risk assessment is usually the toughest subject area. And so, but people need to get better at doing risk assessment and mitigation. And the other thing I will say is that so far, all of our biosafety and biosecurity programs are all focused on life scientists, on biologists or health scientists. We need to bring the engineering community into this, because you will notice that as more and more people do synthetic biology and gene editing systems, etc. For example, if you remember, I told you about the international competition called iGEM. If you look at the teams of iGEM, it consists of maybe 25% to 50% of the team is from the life sciences or biology or biotechnology. The other 50% is from pure engineering, mechanical engineers, materials engineers, etc., who have no background whatsoever in safety, biosafety, biosecurity, or risk assessment and mitigation measures. And they don't even know that these things exist. So we really need to take everything that we have been teaching and everything that we have been learning and expand this. And so for organizations like BRAP, this means outreach to communities that are beyond what the current outreach is. How do you engage engineers? How do you get them interested in these kind of things? These are all challenges that all of us face, and we have to think about it, and we have to come up with solutions. Again, both funding agencies and journals, funding agencies are responsible for funding the research. So they need to make sure there's sufficient review and oversight. Journals, of course, will publish the results, which can be misused, and therefore they also need peer review, policy peer review, and oversight. So Samreen mentioned the 7 areas of identifying threats in research. I think the first 4, the way I have grouped it over here, the first 4 are related to pathogens or toxins. And so if you're doing work with pathogens or toxins, then you have to be careful about those categories, and you have some idea already that, you know, oh, my research could be dual use research of concern. But now we come to these 3 areas that I have highlighted in bold here, which is alter the host range or tropism of an agent or toxin. This can be done unknowingly, and this can be done with this can be an unintended consequence. For example, I showed you the CRISPR chicken or the CRISPR pigs that are being developed for livestock industry. That could be an example of how we are going to change the host range of the agent. This next thing is interfere with the generation of immunity to an agent or toxin. And this is very important because we are going to come up with newer solutions to tackle simple chronic conditions in the body. In the process, we might alter the immune status of the person. And therefore, that person might be far more susceptible to the dangerous pathogens. These are the kinds of things that we have to think 2 steps ahead, and we have to start thinking about what will be the consequences of our research and our applications of these technologies down the road. And the number 7 is, of course, connected to number 6, the susceptibility of the host to an agent. I will also reiterate and focus on the pillars that Martin outlined at the very beginning of the talk. The pillars are still really important. You need to think about outsider threats. You need to think about insider threats. You need to have things like inventory control, information control, personnel control, physical security, transport security, and emergency response plans. All of those things need to be in place. Those are all the same best practices if you are working with synthetic biology and emerging technologies. You need to have all of those systems in place in your lab in order to be able to make sure that you are ensuring biosecurity. With that, I thank you for your attention. And I think now we'll take questions. So I pass this on to the BRAP committee.
Okay, thank you so much, Dr. Prasad. Okay, thank you to all our 3 distinguished speakers. So now we will be reading some questions that are raised by our participants in the chat box. Okay, let me try. For Samreen, sorry. Okay, more or less, there were some common questions for Samreen. Hello, Samreen, Dr. Samreen, can you hear me?
Yes, I can hear you. Yeah.
Okay. I think most of the questions are related to the current pandemic. So we have here some questions from Samarita Lapitan, from Mr. Karandang, Matthew, and Marina Bautista. So more or less, the questions evolve on with the current pandemic of COVID-19. Do you think the COVID-19 pandemic is a product of a laboratory or laboratory grown? So most of the question either can it be a biological weapon, or is it unintentionally released. So they're just asking for your opinion on this matter.
Okay. So we cannot say this because there have been a lot of theories revolving around the social media and internet that this could be one of the biological weapons. I would like to clear one thing, that biological weapon is not only, they're used for different purposes. For example, they're not only to make with high mortality rate, they don't only want people to die. They can be for other purposes. They can be for agriculture, against plants. So these are different types of bioterrorism events. But if we specifically talk about COVID-19, so before there is a proof or evidence that this could be one of the biological weapons or lab-grown virus, we cannot say for sure that whether it is a biological weapon or not. I would like Prasad to comment on this as well.
Thank you, Samreen. I think, as you mentioned, there has been a lot of speculation in the media, and this has been politicized at various levels. But I will draw your attention to a Nature paper. I want to say it was a couple of months ago now, which looked at the sequences very carefully and looked at the evolution of the virus. And they and the authors concluded that in all probability, this is a naturally occurring virus, that it's Mother Nature that produced this virus. And that's how this virus came into the world. And then, of course, it started in animals, and then it infected human beings, and then it spread. There was human-to-human transmission, and now it's all over the world. There is no evidence to suggest that in any way that this was engineered. Because if you look at the other coronaviruses, and if you look at their sequences, and then you look at this sequence, etc., there are ways of telling if this was a man-made modification, and those things are not present in this virus, in this sequence. And so any speculation, there's no basis for any speculation on that. There's also been speculation on whether this was the result of a biosafety incident where there was accidental release. And again, currently there is no information to suggest that that was the case, because at the Wuhan Institute, etc., there was no, people didn't fall sick, and there wasn't a virus that was, identical in sequence, etc. So again, I think as we speak, there is a WHO panel that is currently in China that is looking into this, and I'm sure they will produce a report. But from all indications, this was Mother Nature. I think we underestimate what Mother Nature is capable of producing. And so, this was a bat virus that made its way to humans, and then now it's all over the world.
Okay. So thank you very much, Hamreen and Dr. Prasad. Dr. Martin, do you have any comment on this?
Oh, go ahead, Dr. Jobs.
Okay. So, Samreen, we also have a question here. If you have a research that is sort of to be dual use research of concern, is it advisable to publish it in an open access journal?
It depends upon risk-benefit assessment. Whenever you are planning communication of this type of research, which is a DURC, you always do a risk-benefit assessment before planning your communication. So as per your risk-benefit assessment, you will decide whether you are going to publish it in full, or you want to redact some information, or whether you want to publish it in open access and to everyone, or you want to extract some information. So this all depends on risk-benefit assessment, and this can vary from one research to another. And majority of the cases, the scientific community has supported open publication. Like they supported giving access to all researchers around the world. But definitely it depends upon the risk as well. If the risk of communicating it in open access general is very high, then definitely the communication will be planned accordingly. So there is no it varies from one research to another. So there is no one answer to this question, that whether you will do it always or you cannot do it.
Okay. So thank you, Samreen. So we have here another question for Dr. Prasad from Jed Emil Villavicencio. Questions regarding genetic modification or engineering. So a nation should have a policy and oversight for this technology. Does the world have a governing body for this? Different nations have different agendas, belief, and policy. Is there an international body that can control? And is there a sanction or penalties present?
So currently there isn't a dedicated international body for this. But for example, if there are experiments on humans, that would be under the purview of World Health Organization. Approvals for animals, CRISPR animals and gene edited animals, etc., should, in principle, be reviewed by OIE. And also at the Biological Weapons Convention, which most countries in the world are signatories to, there is now increasing awareness and there's increasing talk about measures that countries should take in order to reduce the risk of this. But to answer your question, there is no single body that is able to review and approve these kinds of things. And it's going to be very challenging to do that, because remember that if you require lengthy review process and approval processes and things like that, that will significantly affect your bioeconomy. And so no country wants to, be disadvantaged when it comes to the economy and when it comes to growing biotechnology. So the solutions will have to be, it will have to be carefully calibrated. And this is one of the challenges that we face.
Okay, thank you, Dr. Prasad. So we have here a very interesting question, which can be answered by Dr. Prasad and the rest of the speakers. Do you think the moral restrictions hinder the progress of gene editing science? What is your personal take about the application of gene editing on humans? Do you approve it? Why or why not? This is from Lee Marvin.
Okay, I can start and then I'll pass it on to my colleagues. Bioethics is critically important. I think that's what you mean by moral responsibility. Ethics is not something that everyone learns in school or in college or later on at the job site. Ethics is also, believe me, I have actually taken ethics classes that have been so boring that, it's hard to stay awake the way that ethics are currently taught. And so we have to make changes. We have to make changes to make sure that people have, when they are thinking about ethics, they have real life examples, real life dilemmas, like the one that Samreen posed, when people did this influenza research and they wanted to publish it. That was also an ethics question. Should that to ask the question, should research be conducted or should research not be conducted? Yes, it's a risk-benefit analysis, but it's also a bioethics analysis. And especially given all the controversies that are taking place around COVID-19, whether it was man-made, whether it was an accident, etc., more and more, those of us who do scientific research will have to explain to our communities what we are doing, how risky it is, and what measures do we have in place to avoid the risk. And so ethics is a big part of this. And so we really have to come together and invest in this. And when it comes to human gene editing, I think that there are two types here. One is germline editing, where essentially you are affecting germline cells, and therefore you are affecting future generations. I am in favor of a moratorium on that. I don't think we are in a place where we should be trying those kinds of experiments. But on the other hand, the example that I showed you, where you can correct a person's vision by taking the cells, modifying, repairing the genetic mutation and putting it back, or doing it in vivo, those kinds of things should be allowed in my mind, because if you make that change, that does not get passed on. That's the distinction I would make. Samreen, how do you see this as dual use research of concern?
I think because their research, there's no one person can master in all types of research, and usually we see that maybe development of code of conduct in each and every organization can help people understand the ethical values, but and what they are needed to do or required to do. But there is also one thing in it, that code of conduct always helps those who want to follow it. It is not for people who don't follow it. So code of conduct also has some limitations. So maybe one thing can be developing a code of conduct in each organization and following it. Maybe you can develop a national code of conduct and then institutional code of conduct for this particular type of research which is being conducted in all universities. Thank you.
Okay. So thank you very much to all our speakers, and thank you for the participants for all your questions. For those whose questions were not read, we will try to collate all your questions so the speakers can address it later. And thank you so much for your active participation during the open forum.
Thank you so much.
So we'll now proceed to the synthesis of the session. So tonight's session is indeed very interesting and fruitful. So I would like to thank once again our three speakers for discussing the biosecurity with the new trends and emerging technologies. So Dr. Moreno, our first speaker, highlights the basic concepts of biosafety and biosecurity, as well as mentioned the milestones of BRAP and PAMET in the last decade here in the Philippines. He also presented the new pillars of biosecurity, emphasizing the dual-use research of concern and emerging technologies as a way of introducing the next two topics of our speakers. So Dr. Samreen discussed the concepts of dual-use research of concerns. She also showed us why is it very important to know about it and how to identify its categories. And in the event that we know we have dual-use research of concern, she also discussed on what to do about it, how to do risk-benefit assessment, and how to develop a risk mitigation and communication and responsible communication plan. And lastly, Dr. Prasad introduced to us synthetic biology and its applications, as well as on how to mitigate the risk of the applications of synthetic biology and how to strengthen biosecurity measures against the new biosecurity threats. So indeed, the three talks are very timely during this time with the current situation that we have. Synthetic biology as well as dual-use research of concerns has its benefits as well as the risks. So our speakers have discussed on what are the risks and the benefits and how to make that. Tonight's session will create a big impact on our own facilities and our own institutions on the way we see biosecurity in this modern age. So thank you very much. But before we're going to end our session, can I ask everyone to turn on your video so we can take a screenshot of all the participants for tonight's session? So may we request all participants to kindly click on your videos just for a moment. We'll take a picture for everyone.